Hi everyone. Up until now, we heard from Sharon and from Daphne about the West Nile virus, the significant or the largest outbreak that we had this year with significant mortality and morbidity. Unfortunately, we also heard from Gili about the possibility of having plasmas with high levels of neutralizing antibodies to reduce mortality. And in this talk, this short talk, I promise you guys, I'm going to tell you a little bit about the West Nile virus monoclonal antibodies that we're trying to develop. So a few months ago, there was a very nice perspective at the New England Journal of Medicine talking about whether it's time to revisit vaccination of West Nile virus, with the idea that since West Nile virus is the leading cause of US arboviral diseases, it's not just in the US, of course, it's also an ongoing public health threat in many areas of the world, including Israel, we just saw this year, but also, and this is, there's high endemicity, and every year this endemicity is going higher and higher in Europe, with outbreaks growing also in that area. Now, it's not just morbidity and mortality, it's also that West Nile virus disease results in substantial cost to patients and society, e.g. an economical burden. Plus, well, we have two great vaccinations against West Nile virus in horses with up to 95% effectiveness. And indeed, when looking at the different vaccines that are currently under development, there are multiple vaccines, including one from the NIH, by the way, which are in different phases, but unfortunately, none of them have yet to mature into a product or even into a clinical phase 3 study. And this is despite the fact that the immunogenicity is extremely good for many of them, and that even, for example, the NIH vaccine, it only requires one dose. When a lot of studies and a lot of thinking is done, that maybe we won't see any West Nile virus vaccines in the near future. And that's because there are really big challenges with West Nile virus vaccinations. The first one is that West Nile virus is really unpredictable. There's a lot of sporadic and unpredictable outbreaks. We all know that from this year in Israel, which we saw the fluctuations in West Nile virus outbreak. We also see that, as you can see here on the left, in the United States, with many areas of the United States changing periodically within West Nile virus outbreaks. We also know that severe disease is observed in older population, and when we're thinking about the trial's endpoints, such as preventing neuroinvasive disease, getting there would take years. And also, again, selecting the geographical areas to where we should pick these for vaccine efficacy trials is very, very hard. It's also a risk, especially for live vaccines, that there's a significant risk for prolonged viremia and adverse events, which is highest in immunocompromised an older population, which is exactly the populations where we would need the vaccines against. With all of this in mind, we thought that maybe monoclonal antibodies could be the answer. And so for monoclonal antibodies, protection is immediate. We think that it may also treat infection even after symptom onset. We saw that again, something like that in what Gili showed just now. It is highly effective against vulnerable population. This is again exactly the population that we would like to give these monoclonal antibodies or these vaccines. There's also a very nice study, a previous study, which was done a few years ago, showing that mice treated with IVIG from Israeli donors were protected from West Nile virus infection. Specifically, I like this study, which was done by a scientist from the Biological Institute, because my father is actually one of the leading authors on this study, but also because it's a very nice study. And for me, it's just amazing to continue this work. And also, as I said, Gili's new data that demonstrates the protective effect of high West Nile virus neutralizing plasma against West Nile virus in humans. So for all of these reasons, we thought that maybe West Nile virus monoclonal antibodies could be a good way to go. And also, it would be great to do it here in Israel. And when you're thinking about it, the adjusted West Nile virus case numbers are highest in Israel. As you can see here, the average or the yearly case numbers, the adjusted case numbers per 100,000 population. And so it's clear that there is West Nile virus is here to stay, and we have it much more than any other area in the world. And also, when looking at this, is again the previous study from the Biological Institute, they showed that IVIG or pooled sera from Israelis, but not from American donors, for example, protected mice from West Nile virus infection. So West Nile virus is endemic in Israel. We know that also from studies which we conducted a few years ago, showing that, again, it's all over the country and with very high seroprevalence of higher than 11% for the total population. So we thought that maybe this could be the real job for starting to do it here in Israel. And we also thought that the SPRI cohort or the Sheba healthcare worker cohort could be the basis for developing this vaccine, this monoclonal antibody, because of all of these wonderful people who we could get the PBMCs from to develop these monoclonal antibodies. And just to remind you, the cohort started from the SARS-CoV-2 Sheba healthcare workers cohort, where Gili started recruiting, and up until now it's about 9,000 Sheba employees. And this has started at the beginning of the COVID vaccine, the COVID pandemic. And this was monitoring how the vaccine works, how infection works, understanding much more about the immune response. And we've done so much work about it that we thought it would be very interesting also to use this cohort in many other developments of either monoclonal antibodies or understanding more about some of the pandemics that we are facing against. And this starts with the SARS-CoV-2 variants, avian influenza, which we all worry about. And just this week, we heard of the first severe case in the United States with avian influenza, the diseases that might come around from the Hajj pilgrims, diseases that are in travelers, such as dengue, Zika, antibiotic resistance, West Nile virus, and now we're also looking at Leishmania. And specifically for West Nile virus, we started this about 2 years ago. The first thing that we've done, and Gili also mentioned that briefly, is that we asked for volunteers from the SARS-CoV-2 Sheba healthcare workers cohort, a little more than 300 volunteers. We received serum from them. We tested them for West Nile virus IgG, and all of the positives, overall there were 14% that were positive, a little more than we anticipated, which was again 11 to 12%, were termed the West Nile virus Sheba healthcare worker cohort. These were then tested for neutralization assay, with the neutralization assay to test them whether they have neutralizing antibodies. About 60% of them had neutralizing antibodies. We then asked for them to give us their PBMCs, which luckily they agreed, and we sent these to the NIH. At the NIH, they picked the 2 or 3 most, the best samples with the highest neutralizing levels. They took their PBMCs, sorted them, and using the rat pig method, which was developed by Dr. Danny Dueck, within a week or so, they were able to produce thousands of monoclonal antibodies, which were then characterized, and this will be the subject of the next talk in a minute by Dr. Ted Pearson, the head of the VRC. So just summarizing the milestones, we started this endeavor at around 2 years ago, in December 2022, with the initial idea of generating these monoclonal antibodies. We then, at the NIH, constructed the West Nile virus probe. We, in the meantime, set up the cohort, tested the sera, and collected PBMCs, sent it to the NIH, which around a year ago started screening for the best candidates, performing the rat pig, the monoclonal antibody selection, and the mice experiments. Overall, as I said, this took 2 years. Unfortunately, we're not able to reach the milestone of last year, which was wonderful if we could, but we weren't in that stage. However, now we know that in cases of urgency, we really could narrow all of this into a few months. With this, I would like to end and give the realm to Dr. Ted Pearson, the director of the VRC, who will talk about both the monoclonal antibody selection and the mice experiments. Thank you, guys.